Hello and welcome to the World Molecular Imaging Society live webinar today, presented by the Educational Committee and the Imaging in Cell and Immune Therapies Interest Group, also known as ICIT. Moderating today, we have Dr. Gilbert Fruworth, who's a reader in imaging biology at King's College London, and myself, Israt Alam, senior scientist at Stanford University. And together we co-chair ICIT. So the aim of this interest group really is to improve the monitoring of molecular and cellular immunotherapies. And we do this through identification of new biomarkers of immune response, but also through advancing the implementation of imaging technologies globally. We try and achieve this by facilitating dialogue and cooperation between scientists in academia, in industry, but also we oversee knowledge transfer and education. So today's webinar is specifically tailored to convey the technicalities when employing reporter gene methodologies, which have been absolutely central to cellular immunotherapy tracking and imaging. We are thrilled to have with us three fantastic speakers and experts in the use of these tools: Dr. Georgia Zambito, Sean McRae, and Dr. Rafael Sanchez-Pupo, who will share their knowledge of important in vitro and in vivo considerations when employing these techniques. Please note that this webinar is being recorded and will be sent in a follow-up email after the webinar's done, and also be posted on the WMIS website. If you have questions for us, please do enter them into the live chat or the Q&A bubble, and these will be addressed after the two talks at the Q&A. Please also join us following the webinar. We will also be having a social lounge, which will give you the opportunity to further interact with our speakers today. So I will start off by introducing our first speaker of the day, Dr. Georgia Zambito, who's a postdoctoral researcher at the Erasmus Medical Center. And she's an expert in preclinical optical imaging. She first joined as a PhD candidate in 2016, and under the supervision of Dr. Laura Mezzanotte, trained in bioluminescence techniques for in vivo T-cell tracking. She's currently working on the development of a microfluidic chip and expanding the toolbox of bioluminescent reporters for non-invasive analysis of inflammatory and mechanotransduction biomarkers. She's also a committee member of the Dutch Young Molecular Imaging Community. So Georgia will be sharing with us practical insights, recommendations, and pitfalls related to the use of bioluminescent reporters. For our second talk, we'll be having two speakers, the first being Sean McRae, who's a graduate student at the Robarts Research Institute, the University of Western Ontario. Sean graduated with a bachelor's in medical and biological physics. He's currently a third-year PhD candidate working under the supervision of Dr. John Ronald and Dr. Timothy Shaw. And he's doing some really exciting research which focuses on tracking delivery of CAR NK cells and monitoring therapeutic response using multinuclear magnetic resonance imaging in preclinical models. So Sean will be joined by Dr. Rafael Sanchez-Pupo, who's a postdoctoral researcher, also at the Robarts Research Institute, and he's an expert in protein chemistry and protein engineering. Rafael graduated with a bachelor's in biochemistry and molecular biology from the University of Havana, and then obtained his PhD in cell biology from Dr. Sylvia Penuela's lab at the University of Western Ontario. For this, he received the Keith Elmore Doctoral Achievement Award, which is awarded to the top PhD student. So currently, Rafael is also in Dr. John Ronald's lab, where he employs CRISPR-Cas9 genetic editing to create tools for monitoring CAR T cells using optical MRI and PET imaging in preclinical ovarian cancer models. So Rafael and Sean will be sharing insights on practical considerations for the use of MRI reporters for immune cell tracking. So without further ado, I'm going to hand you over to our first speaker, Dr. Georgia Zambito.
Yes, I'll try. Thank you, Isra and Gilbert, first for inviting me as a speaker. I'll try to share my screen. Almost there. Can you see my screen?
Yes, Georgia.
Okay, thank you. So hello everyone. Today I'm gonna talk about how to target cells with a bioluminescent reporter gene and how to enhance the light signal for in vivo bioluminescence imaging. I will try to give you a very practical talk and some suggestions, some advice how to perform the bioluminescence imaging in vivo. So first of all, just roughly, I would like to mention what's the bioluminescence. So bioluminescence, it's a biochemical reaction where a luciferase enzyme can oxidize a luciferin substrate, and the binding of the enzyme with the substrate generates light. But here you can see like how more or less it works, the pipeline. So you have your luciferase vector of choice. You can transduce your cells and the cells can emit the light. Then you expand the cells of interest, and the cells can be injected in the mouse. Perhaps the mouse is bearing a tumor model that you are following, that you are monitoring. So by injection of, in this case, the luciferin as a substrate, you can image the mouse over time and also follow the tumor and the tumor growth and so on. So, and be ready for the next experiment. But the first question, if you start an experiment on bioluminescence imaging, is regarding, is first of all, what kind of luciferase reporter you have to use, and what kind of substrate you have to use. Because perhaps you know that now in the bioluminescent field, there are many engineered luciferases and also many synthetic luciferin substrates that have been used and created to improve the performance of those systems. And here you can see, by using those different systems, you can improve your outcomes and improve, for example, sensitivity, pharmacokinetics and pharmacodynamics. And also this gives the possibility also to have a multicolor bioluminescence imaging in vivo. Of course, you have also to think and that the bioluminescence can be affected by, for example, hemoglobin. So because of course it's subject to optical, of course it's regarding emission of the light. So you have to think that if you want to image in surface or in deep tissue, hemoglobin can affect your imaging session. So, for example, you can see here that for a surface imaging, you have the hemoglobin that can affect your imaging, but for a deep tissue imaging, so ranging, for example, from 650 and 900, of course, the hemoglobin has a lower impact on your imaging session. And so that's also very important to understand at what is the depth of your imaging and what you want to track and what you want to monitor over time. And if it's first superficial and then it goes in depth, or if it stays superficial. So you have to think and take into account this kind of settings. And speaking of superficial imaging, To my knowledge, you can, of course, have mainly two different luciferases. You can have a terrestrial luciferases that they are depending on ATP, and the marine luciferase that they are ATP independent. So mainly you can distinguish them with that. And among the terrestrial luciferase, you can have, for example, CBG2, which is a click beetle. There is also CBR2. Those click beetles are have been in the field, have been mutated in order to have a distinct colors in vivo. For superficial, I advise you to use a CBG2 with a D-luciferin substrate, because it gives a very bright emission of bioluminescence, and still it's under 600 nanometers, but still it's a good combination if you want to image superficial cellular events or molecular events. And then you have the F-look and the other derived mutants. Of course, you can use F-look with the D-luciferin, which is also a good emission, which has also good emission, but of course you cannot use for brain imaging, for example, because of the permeability. That it's a bit poor, but if you want to image the brain, for example, Seq-look has been synthesized just for that purpose, and can be used for brain imaging. Amongst the marine luciferases, you can have nanolock. I think it's quite popular now because because with the nanolock you can expand really the applications now. And the nanolook can be combined with a colenterazine-based substrate. It's called the furimazine, and the emission of this system, it's in the blue light. So it's just for superficial imaging. I won't go for a deeper imaging purposes. And also in this case, other substrates have been created, for example, fluorofurimazine, hydrofurimazine, that makes the substrate more soluble and more stable over time. And they have a very also bright emission of light, and can be really used for this kind of superficial applications, but also with a high emission of photons. And the nanolook has also been used for the breath technology. And here I mentioned Antares, but we have also nanolantern, and they can be, because of the breath technology, they can be used coupled with different fluorescent proteins, so you can have different colors. And then if you want to go in deep, I, for example, often use click beetles, because we have in our group developed CBR2 and CBG2, and combined with an naphthyl luciferin substrate, which is quite stable and soluble, and you can go far in the near infrared region of the spectrum. So that's quite convenient also for a deep tissue imaging. And but nevertheless, for example, has been created the infraluciferin, which is also very used for example, tracking cells. I will give you an example later. And it also goes to up to 650 nanometers, so also for deep tissue imaging. And then Acalumin HCl, the ACA-BLI system that is called, and ACA-BLI, and that's been optimized also for tracking cells and also deep tissue imaging. And here, I mean, we are not in the deep imaging, but I wanted just to mention that recently a new substrate has been created, which is the cephalofurimazine, which can be coupled with a nanolook, and gives also very bright emission of photons, and is permeable. It's a substrate that can be used for imaging the brain, so has a very good blood-brain barrier permeability. So once I gave you this overview of the substrates and luciferases in the field, so if you have in mind, for example, to use for example, you want to track cells, so you want to exclude, for example, blue emitting luciferases, you want to go for a red or near infrared luciferases. So let's say you want to use this kind of systems. After selecting these systems, and perhaps you want to try, I don't know, AKBLI and some F-look mutants and the click beetles, then you have to choose the promoters that are needed to make your lentiviral vector, because usually we have, for example, these promoters, the CIG promoter, EF1 and PJK, but for example, for transducing T cells, for example, to monitor and tracking them, EF1 has been resulted the most effective one. So we are also using a EF1 promoter, which is also very, to say, effective for cell transduction and to make the transgene working. So I would go for EF1 as well. Of course, it depends if you want to track T cells, you can use other promoters, which are also very convenient and useful, for example, with the human CD2, or if you want to detect, for example, T cells, you can use a promoter for CD4 or CD8 promoters, or if you want to follow macrophages, you can use the CSF1 or arginase for tumor-associated macrophages. So once you have also selected your promoter, if you, for example, go for a simple and standard promoter that is the EF1 promoter, then I advise you to design your construct, your lentiviral construct by adding a GFP, for example, a fluorescent protein that can be used to sort your cells. Because once you transduce the cells with your lentiviral vectors, and of course you want to select the positive ones, and the GFP can be used as a positive control that can be used, for example, in for the cytoperimeter to select, to sort the positive cells from the negative, and then you can expand the positive cells. So once you expand the positive cells, I usually proceed with in vitro characterizations of your systems, because perhaps you have selected 2 or 3 systems, but still you would like to check, of course, before going in vivo, you would like to check how they really work in vitro, and what is the best substrate to use for your in vivo application. So first of all, what I usually do, I check the performance of the systems in a live cells. So the positive cells that I have sorted, I plate them in a black 96 well plate. And here I give you, for example, here, for example, you can make a kind of a standard curve with the different concentrations of cells, and you can use also different substrates and check the photon flux of the systems, of the luciferase systems you are testing, and this will give you already an idea if it's a very bright system, that it can be used in vivo, for example. And here it's also another imaging where you can, for example, test the different luciferases with the different luciferins and see if you want do multicolor imaging, if this can be done by crossing the substrate, not by crossing, but by using the different systems, and see if the level of photon flux is also comparable. Because if it's not comparable, I won't go in vivo. Otherwise, you can have false. Your results can be affected by the already differences in the photon flux. So once I have evaluated the performance of the systems in vitro, you can already make a screen of the systems that in theory can work in vivo. What we do is if for example, the system has a photon flux that is lower, for example, than 10 to the 4th photons per seconds, I won't trust it for deep tissue imaging, for example, or to track cells. Maybe can be too low than in vivo. But in the first thing that once you select your systems that can be used in vivo, the first step I would do is related to the signal background of your substrate, because there are some substrates that can give you, for example, some background. So it's good to have that in mind, especially when you have to quantify and to monitor and follow your cells over time. For example, here you can see that when you inject only D-luciferin, it doesn't have a background. When you inject a furimazine or the other analogs, they have a little background in the bladder. And when you inject acalumine alone, you can see some hepatic signal as a background. So that's something that needs to take into account when you perform your in vivo experiments. And of course, the injection of the substrate can change your slightly change your decision also on the system to be used, because, for example, if you go with intravenous injection of the substrate, of course the sensitivity goes higher, but if you want to have repeated imaging sessions in a short period of time, then I advise you to go with peritoneal injection of the substrate. And speaking of, for example, multicolor imaging or dual color imaging of mice in vivo, applied in vivo bioluminescence imaging, you have always to compare the spectrum of your luciferase-luciferin couple that you want to use, because maybe in vitro the two spectra that you see here, they can be well separated. So you might think that by using your filters at the IVIS machine, for example, you can separate and distinguish the two cell populations later in vivo. But it might happen that in vivo, this, especially for the green and blue light-emitting luciferases, the spectra emission can change a bit, can shift a bit, and then maybe you won't be able to separate the two cell populations anymore. So I would suggest you to, of course, check it in vitro, but of course also to check again in vivo, because when you do the spectral mixing, for example, when you do, for example, dual color bioluminescence, you need a spectral mixing. If you use, especially if you use a system with the same, for example, a system in which you want to inject the same substrate and identify two different populations. And in that case, I suggest you to use the spectral mixing algorithm that is very easy to do at the IBIS spectrum. And once you inject your population, the two populations, for example, emitting here, you can see the example of the orange and the red light, you can image your mouse, and then you can collect the images by using the different filters that they are installed in the IBIS spectrum, and the algorithm will be able to separate the light and can distinguish the population located also in the same area in the mouse. So, for example, you will have orange for CBG2 and red for CBR2, and the algorithm will be able to distinguish the two cell populations. Then I'll give you a few examples that I have taken into account when I was performing this, some of my experiments in Vivo. And these are mainly to track immune cells in general. So I started, for example, reading a paper written by Kontag in 2006. In this paper, I think it's a really nice paper to read because it gives you the right step to to organize and design your experiment from the in vitro part and the characterization of your cells and of your luciferases, luciferase expressing cells to the in vivo applications. So in this paper, for example, they were labeling the cells from extracted from a transgenic donor mouse, and the cells were transferred to an unlabeled wild-type recipient mouse, and then the reagent was added, the substrate, and then they were imaging the animal and localized the cells. So here, as I suggested you before, what they did was first to check in vitro how much was the photon flux the system, the luciferin system they selected, how much was the photon flux emitted by this system per number of cells. And like that, they could understand if already the system was feasible to use in vivo. And here you can see the difference when they were injecting different number of cells in the mouse. And here you and then after doing all these characterizations of the system, they decided to go in vivo, where they have transplanted cells, T cells, and they were comparing the syngeneic recipient and allogeneic recipient, and they could follow the T cells and monitor them over time and follow them in the mice. So that was already something already used with a D-luciferin and LUC2 combination system. Okay, and here is another pioneering, let's say, paper on a multimodality imaging of T-cell hybridoma. And here, of course, it's one of the first papers. Of course, also, if I'm not wrong, also Ponomarev started with a multimodality imaging of cells as well. I'm here showing one example in which they could, for example, use a multimodality imaging setting in which the lentiviral vector was able to express probe for PET imaging, but also a probe for bioluminescence. And here I wanted to show this case because for bioluminescence, they used Renilla, that is a luciferase, a blue emitting luciferase, and you can see that it was in a black fur mouse. Of course, was not able, I mean, the authors were not really able to see the bioluminescence in the mouse because it was blue emitting, so the light was completely absorbed also by the black fur of the mouse, but it was visible in the paws of the mouse, where they also wanted to check T cells migration in the paws affected by arthritis. And going further, this is another example where T cells were transduced with an optimized firefly luciferase, this EF look, because of course over time there are always new technologies and new way to engineer the cells and emit much more light, as you can see here. And in this paper, they were able to see fewer than 10 cells by bioluminescence. So that's just an example to show you that can be very sensitive also in vivo in a furred mouse. And talking about tracking, for example, CAR T cells, in this paper written by Stolf, they could track CAR T cells by using an infraluciferin substrate. As I told you at the very beginning, the infraluciferin has been optimized for emitting in the near infrared region of the spectrum, and they could make a nice, how to say, system with the F-look mutant and the infraluciferin substrate. And here they applied the spectral mixing because they had the tumors emitting, let's say, green light, and the CAR T cells emitting red light. And by applying the spectral mixing, you can see here in the mixing green that they could check the tumor status, for example, here, and in the mixing red, they could track the CAR T cells injected and homing the tumor side. And when you do the spectral mixing, the algorithm is also able to give you a composite where you can distinguish the green from the red in this case. And they could follow also the CAR T cells post injections, and they detected them for 6 days post injections of CAR T cells. So that's quite also important to monitor the dynamics and the behavior of CAR T cells after injections. And that's also another example of dual color bioluminescence imaging. And in this case has been done with an orthogonal systems, with the two orthogonal systems, because here we have Antares, so nanolook expressing nanolook used for a breath, for the breath technology, in which has been coupled with the CIFOP orange fluorescent protein and used with fluorofurimazine, which is a analog of furimazine, and here they used the Antares was used to track the tumor and the tumor growth over time and the tumor cells, and then they injected the CAR T cells expressing the ACA-BLI system, so the ACA-look plus acalumin, and they could also detect the CAR T cells over time and actually 30 days after injections of CAR T cells. So when you use these orthogonal, these kind of systems for a dual color, you have basically two choices. Either you use two different luciferases and you inject the same substrate, or you use two completely different systems in which you have to inject two different substrates specific for the luciferases, for the luciferase selected. And I have the last example. In this example is made by the group of Gambir. Here they could detect macrophages, and they could use tumor-associated macrophages as a sensor in acute inflammation. And I really like, I wanted to show this example because I really like how they designed the experiment. So they engineered the macrophages, and these macrophages have Gaucher. So Gaucher is a luciferase that is mainly secreted in the blood. So after injection of the macrophages could migrate to the site of inflammation, and then by isolating the blood, they could detect the tumor-associated macrophages or the inflammatory macrophages at the tumor site. So it's quite nice as a sensor to detect also small metastases in the body. And with that, I hope I'm on time, and I would like to thank you all for the attention and all the organizer for this nice webinar. And if you have further questions, please write on the chat, and I will be also available in the social lounge if you want to discuss later. Thank you all for the attention.
All right, I'll pull up my slides here. Can you just make sure that you can see my screen and you can hear me? Good to go. Awesome. All right. So today, Raphael, my colleague, and I will be talking about exploiting gene reporters for in vivo cell tracking using magnetic resonance imaging. So just to give a quick overview of what we would like to cover in today's webinar, I'd like to talk about the role of MRI in molecular imaging. I want to discuss some MR basics as well as different contrast mechanisms. I want to talk about some MR reported genes specifically, giving examples. Then I'm going to talk about some practical imaging considerations. Then I'll hand it over to Raphael, who's going to talk about some experimental methodology using examples from the literature. Okay, so let's begin by talking about the role of MRI in molecular imaging. So we just heard from Georgia a really nice talk on how bioluminescence imaging can be used for reported gene tracking of immune cells. And along the translational pipeline, typically bioluminescence is very useful at early stages before we scale up experiments to a clinically relevant imaging modality such as positron emission tomography. The reason that PET is used very commonly is because it is very sensitive. And when we're talking about immune cell tracking, we're trying to image small groups of cells in living systems. So really sensitivity is what will kind of get us there. So then where does MRI fit into this pipeline? Well, the goal of this webinar today is not to try to convince you why MRI is superior, but instead what we're going to try to do is convince you why MR provides utility along this pipeline before leading into PET experiments. So how do these two imaging modalities stack up against each other? Well, if we compare things like availability, relative affordability, logistical complexity, sensitivity, the ability to generate quantitative maps, the ability to generate different contrast parameters, as well as spatial resolution, we can see that, in fact, MR does have utility over PET in some cases. But the main difference between the two is really the sensitivity. And when we're talking about probe sensitivity, we are looking at differences in several orders of magnitude between MR and PET. And what this means for MRI is we're needing to give very high doses of different metals, such as gadolinium or iron, to be able to overcome this limited inherent sensitivity in order to track our cells. And this is something that has implications in the selection of which reporter gene you're going to use for your experiments potentially. So we'll come back to that later on. But before I do any of that, I just want to bring everyone up to speed on some MR basics, just to make sure that we're all kind of on the same page. So an MR scanner consists of three different sets of magnets. We have the main magnet. So this is, if you have a 3 Tesla scanner, then the strength of your main magnet is 3 Tesla. This is what's responsible for magnetizing the sample when you put it into your scanner. We then have gradient magnets. These are responsible for spatial localization. Then we have RF coils, which have 2 different roles. First, they excite the spins within a sample, and also they can actually detect the sample upon relaxation. So I want to talk a little bit more about the role of these RF coils. So looking at RF excitation first. So I want to draw your attention to this blue vector that's pointing along the z-axis. By convention, the z-axis we say is along the bore of the magnet. And what happens is when you put something in the scanner that is rich in protons, each proton will act as a tiny bar magnet having its own magnetic moment. And the sum of these magnetic moments leads to the magnetization vector that is shown here. So in its low energy state, what it's doing is it's processing about the z-axis. Once we apply an RF pulse, it's going to move from its low energy state to a high energy state. And we can see here that it processes down into the XY plane. And it will stay there until the B1 field generated by the RF coil is turned off. So let's look at that scenario. So when we turn off the B1 field, we get relaxation. So the vector is now going to go from its high energy state back down to its low energy state by processing until it is fully magnetized along the z-axis. So if we break up this relaxation into two components, we can look first at the regrowth of this vector along the z-axis. And this is what gives us our longitudinal relaxation, also called spin lattice relaxation. And the time constant that we use to describe the regrowth along the z-axis, we call T1. We can also report this in terms of a relaxation rate, which is simply the inverse of T1. Next, if we look at the component of shrinking in the XY plane, we get the transverse relaxation, also called the spin-spin relaxation. And this relaxation is governed by a T2 time constant. And once again, if we take the inverse, we get a relaxation rate. So sometimes we use T1 and R1 interchangeably, so just something to be aware of moving forward in the presentation. So T1 and T2 are really fundamental to MRI, as these properties are tissue dependent and are very useful in generating different types of contrast in our images. So now I want to talk about MR contrast mechanisms. So the most fundamental type of MR contrast is called nuclear density. So when we're talking about proton imaging, a nuclear density weighted image can show us essentially a concentration map of where protons are located within our field of view. So this can be of use in some cases, but as far as reporter gene imaging for immune cell tracking, we're actually more interested in parameters like T1 weighting and T2 weighting. And these are sequences that really bring out differences in these T1 and T2 properties that I just mentioned. So certain other reporter genes actually use specialized types of imaging contrast, such as CEST and diffusion weighting. Unfortunately, we don't have time to get into those today, but just something to be aware of, that we can get multiple kinds of contrast from our MR images. So now let's talk about what MR reporter genes actually exist. So what we did here is we took examples from the literature and we tried to classify them based on their contrast type that they generate, as well as their substrate, and also whether or not they are enzymes, transporters, receptors, or whether they fall into the other category. So unfortunately, again, we don't have time to go through all these examples today, but something to note here is that a good chunk of these MR reporter genes actually rely on T2 contrast. And if you recall back to the beginning of the presentation, I mentioned that when we use MR in both reporter gene settings and in the clinical setting, sensitivity is really kind of what limits us. But when we use T2 as our contrast mechanism, when we supplement with iron, we can actually cause dephasing of the local spins, actually giving a higher sensitivity to detecting cells when we use this T2 contrast. So we're going to be talking about two specific examples today. First, we're going to mention the organic anion transporting polypeptide 1. And this is a surface receptor that sequesters amphiphilic contrast agents for T1 signal enhancement. This is the MR reporter gene that we use in our lab. To date, we've mostly published studies that use this transporter to track cancer cells. But we're also trying to move into using this towards tracking things like CAR T cells and CAR NK cells. We're also going to talk about ferritin, which is an iron storage protein that leads to T2 reduction and signal loss. So when we looked into the literature and tried to find studies that actually are implementing MR reporter genes for immune cell tracking, most actually do use this ferritin reporter based on the increased sensitivity that I mentioned before with iron-based MR reporter genes. Okay, so before Raphael talks about some specific examples of methodology behind using MR reporter genes, I just want to talk about some technical considerations. So anyone who's doing MR research should be familiar with what SNR and CNR are and how they kind of affect how we plan our experiments. So first, let's talk about SNR. So SNR is the signal to noise ratio, and it is said that a good SNR value is necessary for the clear delineation of features in a region of interest within your image. So what does this actually mean? Well, if we look at the brain image on the left there, we see that this image has high SNR, and it's quite easy to tell apart the differences kind of see the tissue differences within the brain. Now, if we look at the image on the right, this image has a low signal to noise ratio, meaning that there is a lot of noise, making the image appear very grainy, and it's harder to kind of pick out those fine details that we're after. So high SNR is very important to our image. What else is important though? The contrast to noise ratio is also very important. So kind of extending SNR a little bit here, similar idea, but for the contrast to noise ratio, we need for two ROIs to be distinguishable, we need a very good contrast to noise ratio. And this is really taking differences in signal intensity between two different types of tissue and then normalizing by the noise. So the key to unlocking sensitivity in your experiments that aim to track immune cells will be maximizing your SNR. So I'm going to try to talk about 3 technical considerations in your imaging setup that can lead to increased SNR. So first, let's go back to talk about the B0 field strength. So if you recall, this is the main magnetic field that I was mentioning earlier. So it turns out that SNR actually scales linearly with the B0 field strength. So if we work at a higher magnetic field, we should be able to pull out more SNR for our image. And the consequence of this is we can actually end up imaging at a higher resolution, decreasing our voxel size to try to really pull out small tissue changes in whatever system we're imaging. So this can actually be something that's very useful. But just something to be aware of, as we change our magnetic field strength, we also get differences in T1 and T2. So the example I'm showing here, this is showing the change in relaxation rate in the R1 relaxation rate for different tissues in mice and how they change at field strengths leading up to 3 Tesla. So we see that T1 values actually scale with B0, whereas T2 values actually go the opposite way. So with increasing field strength, the T2 values actually decrease. So another thing to be aware of, though, is if you're using an exogenous tracer, some probes have different field strengths that may not be acting in the same way as the tissue changes. So for example, if you look at a property known as relaxivity, which is little r1, not to be confused with capital R1, which is a relaxation rate. Relaxivity is a measure of a probe's ability to influence T1 and T2. And the examples that I'm showing here, we have 2 gadolinium-based probes that have decreasing relaxivity as you increase a field strength. Whereas I'm showing a manganese-based probe here in red that as the field strength increases, we actually get a higher relaxivity. So it's just important to kind of understand the differences in your tissue, but also your probe. So what field strength should you work at then? Well, this depends on many factors, but it's really important to use what you have available to you as a start. So another consideration that you want to make is in what pulse sequence you're going to use for your image. So pulse sequences are the sequence of RF and gradient events that lead to the formation of an image. So it's essentially a roadmap for when things are being turned on and turned off. So how do we choose the right pulse sequence for our experiment? Well, we have to decide the type of contrast that we're after, whether or not we want T1, T2, that kind of thing. Do we want to use a spin echo pulse sequence? Do we want to use a gradient echo pulse sequence? This can have implications on the scan time of our experiment. Are we concerned with the power deposition from repeated RF pulses, that kind of thing? So in the amount of time that I have today, I won't be able to go through all of these examples, but I've just listed some here depending on kind of what contrast you're after. These might be good pulse sequences for you to look into. But really what's key here is the way to maximize your SNR is to actually look at the signal equation that's governing the pulse sequence that you choose and be able to pick out the optimal TR and TE to get the best contrast based on your expected T1 and T2 values. So parameter optimization is very important in MRI, and it's also very important to have expertise. So personnel that can help you do these to really get the most out of your images. So another special pulse sequence that I want to talk about is inversion recovery. So in our lab, as I mentioned, we use the OATP1 reporter that takes up a gadolinium contrast agent. So we're interested in looking at differences in T1. So rather than just looking at signal intensity in an image, using mapping techniques, we can actually pull out the T1 and T2 properties to be able to quantify, how we're detecting changes in those properties. So using inversion recovery, essentially what we're doing is we're administering a 180-degree RF pulse followed by a 90-degree RF pulse, and we're spacing those pulses by what's called an inversion time. So you can see here in the top right corner, if we take images at different inversion times, we get images with different contrast. And using a little bit of math, we can actually do some curve fitting to pull out the R1 value on a pixel by pixel basis, which can really help us do in vitro reporter gene function validation to see if our OATP1 reporter is working. So this is a very useful tool in vitro, as I mentioned. If you want to do something similar in vitro, In theory, you could use inversion recovery, but what you'll find is it actually takes a very long time. So in the curve that I'm showing on the left-hand side here, I'm showing 15 different inversion times. So each of those images would have taken about 4 minutes to acquire. So this may be a longer time scale than what you're able to do for your in vivo experiments. So other techniques exist, such as the variable flip angle or despot1 sequence, in order to perform faster in vivo mapping. Okay, so the final technical consideration that I want to talk about is in radio frequency hardware. So that final set of magnets that I mentioned earlier, the RF coils. So they're responsible for generating the B1 field as well as detecting signal upon relaxation. So it is really important that you're choosing the right coil for the right experiment, and this will change based on the anatomical region that you're trying to image as well as the animal model that you're working with. So there are a lot of considerations that go into choosing the right coil. Unfortunately, I don't have time today, but what I will do is point your attention towards a really good review paper called RF Coils, A Practical Guide for Non-Physicists, and this will cover everything you need to know in choosing the appropriate coil for your experiment. But relating this back to sensitivity, one thing that I do want to point out is tuning and matching is very important. This should be done before each imaging experiment to really be able to have reproducibility of the sensitivity that you're getting from day to day. So it's really important to be tuning your coil to the Larmor frequency as well as matching impedances to get the most out of your experiments. So as you can see, there are a lot of technical considerations that go into MR imaging, but hopefully that doesn't scare anybody. It's just kind of the reality of doing MRI. So now I'm going to pass it off to Raphael, and he's going to talk about some examples from the literature. So I'll stop my share here.
Thank you very much, Sean. I hope that you guys kind of got the idea of what are the technical hurdles to you have to face to do these kind of experiments with MRI. But at the same time, I hope you guys got some sort of excitation with it, because it's actually a really cool technique that when I came to the lab first time was mind-blowing to me, at least. And I will just start, I have limited time, but I just wanted to point out some of the details that experimentally we have to keep in mind when we're doing experiments using MR reporter genes. So in this case, we just go back to the question, like which report gene it's gonna be right for you. And as you just saw from Sean's presentation, it's usually depends on starting with what kind of contrast you would like to acquire, whether it's a positive contrast or a negative one. What kind of, that's for the conventional MRI, but maybe you want to just go further and look into specialized MRI. And for that, as the table we listed, there are certain reporter genes that are specific. Another aspect for this selection, it's kind of tracer or contrast agent you want to use. In this case, on some of them, like ferritin, can even use the endogenous pool of iron from the body. That will be an easy assessment if you don't want to introduce anything into the experimental model you want to do. But sometimes, which might be better, you can administer exogenous sources of that. And even though it will vary depending on the reported gene to use. This is also the issue of reversibility. This, depending on the reporter gene you're using, if it's not enzymatic, it's transporter, usually these contrast agents actually go in the cell and then go out at the same time. So there's an issue of the timing in which this will happen, and you have to consider that. There's other aspects which the translatability, whether you're just going to stay preclinical or clinical. For clinical, from the preclinical point of view, you probably have to use one that matches the host, for example, in which you want to try to do your experiments, and it should not do anything wrong in the body in that case. There are actually consideration of the cell type, as you saw from the previous presentation from Dr. Zambito. Depending on this immune cell type, you might select conditions that prefer or favor the expression of that particular gene in that cell type, but at the same time, because that's not usually a normal gene that is expressed, it might have consequences to the cell itself. So that's why you do the in vitro analysis of the inclusion of reporter genes. The consideration of immunogenicity goes well with the translatability of it. Anything that is immunogenic it won't be translatable at all. as well as the consideration of whether you want that gene constitutively activated and constantly being expressed, which increments the chance of seeing the signal all the time, or just you want a system that is inducible or activatable. And finally, the consideration of adding multimodality to your experiments. Sometimes you can use a reporter gene that can actually give you multimodality itself, like OATP will be a good example for that. But sometimes it's better just to combine with different other genes from other modalities to increase the chance of success in your experiments. So at the end, it all depends on the goal you're gonna do, and it must be determined experimentally. So as you have seen from the BLI session and MRI session, and basically the general workflow starts with the reporter gene selection, moving into the cell engineering with reporter gene, which I won't get too much into details. Dr. Zambito explained really well that part. But once you got the cells and reached the pool of cells that have reported gene, you proceed to move into in vitro experiments just to validate that the reported gene is working as intended. And at the same time, look at what are the changes that might be causing to your cell system. In this case, could be assessment of measurement of proliferation, migration, phenotypic assessment of the cells, because that will impair or not impair, but influence the actual experimental question that you might have, as MRI will be the imaging tool you want to use. And then when you move into the in vivo experiments, there you just will have to set a set of experiments in which will help you to determine sensitivity of your technique, the construct agent dose that you're going to use, the clearance and kinetics, pharmacokinetics of it, as well as to determine what's the best imaging window and intervals in which your experimental setup will be placed. And of course, it requires the whole optimization of image acquisition and the analysis after. So in the final step, it usually comes with a validation step. That's if it's in the preclinical side, you can do ex vivo analysis using histology or microscopy to analyze the tissues and use orthogonal techniques that will confirm the findings you find in the in vivo imaging. So in the sense of what are actually the experimental considerations we have to look into now that we know the technical part and the pipeline, I will say everything starts with the kind of type of contrast. It's generally seen that when you see increasing signal, it's even easier to detect any changes. And this is the case, for example, for OATP experiments in which OATP expressing tumors can be differentiated from those tumors that do not have a reported gene. And in this case, doing T1 weighted imaging, you will be able to see after contrast the increase in signal or in positive contrast that those yield. But in contrast to other techniques like doing T2 weighted imaging, using, for example, ferritin, in this case, you will see that cells that once they accumulate iron inside, what you will detect is a decrease in signal or negative contrast, which can blend in with regions of the anatomical regions that actually have a darker appearance, and it will be hard to tell when the changes is occurring. That's especially true when you are looking into tissue interspaces. But also I want to highlight that every of these experiments always require you to have naive cells or non-engineered cells as a control to see the basal level of uptake of the contrast agent or any other inferences. So in conclusion, what I want to say here is that if you use the increased signal approach, that will be easier to quantify. However, the new developments of these report genes techniques have already shown that, for example, for ferritin that you usually use to do the T2 weighted images, ferritin also has the capacity of bind manganese metal. So in that case, if you will to do a T1 weighted imaging using manganese supplementation, there is this nice paper here showing, and you can look it online, how the author were able to show that at least you can have a version of bright ferritin using the heavy chain of ferritin that actually shows increasing signals in regions of the body that cells expressing ferritin were, that you could not see with the normal iron supplementation. And this actually awaited, it was even better performing than other reporter genes like the MT1 that also it's transporter for manganese ions. So in this case, the authors show nicely that in a single dose of manganese into the animal, for up to 5 days, it will be able to image significant differences in the R1 relaxation rates. So another of the details I want to talk about is the sensitivity. Every time you engineer cells, you will have to know that within the system, you're able to detect them even at low levels. And that's especially important for experiments involving immune cells. And I would say that broadly, the sensitivity will depend on the reported gene expression level, the number of cells that are actually expressing that reported gene, and as well as the signal background, as Sean talked about, and the contrast to noise ratio. And in typical experiment, for example, with OATP, we use non-transduced cells compared to reported expressing cells that in vitro are incubated with the contrast agent, in this case, Primovis or gadolinium agent. Then you wash the cells, and only the cells you load them into the phantom models and do the imaging. The detail here is you want to decrease the number of cells, so you will be able to see decreasing signal per voxel or per pixel, and that will give you a sense of how many cells you can see at the minimum of signal level, threshold level. But the problem is if you don't include, for example, other cells like combining cells that won't have the reported gene, you might not get an accurate assessment of the sensitivity of your system. So it's sometimes best to have a mix of cells that you know exactly which cell it is, how many of them, proportions, to assess better the sensitivity of your system. And here, this example, what I'm just putting here, is even the same amount of cells, but in different distribution within the box cell might give you differences in signal detection. And this is an example from our group that using I1 mapping, the authors were able to see that what proportion of cells were actually significantly different compared to the naive or non-engineered cells, as well as including controls for negative control for the contrast agent to estimate the background. Moving to in vivo studies, it's the similar manner. You use different amount of cells, you implant them into the mouse, for example, and then you measure the signal in it. And if you calculate the CNR, the contrast to noise ratio in those regions, and you compare them, you will kind of able to see like what's the minimum amount of cells that still give you a confident feeling of sensing significant difference between engineered cells and non-engineered cells. And usually you should have both. And this is also showing that the multimodality also helps for this, because these authors use BLI as a way to assess in parallel the sensitivity of the system. Another way that I want to talk about is using preloaded cells. Preloaded cells with the contrast agent is kind of the idea of, in the best conditions, what's the best uptake you can see on the cells once they are injected into the animal. The issue here is this is basically you are forcing the agent to be in the cells, assuming that's the perfect uptake. And it's not as comparable exactly as this model in which you use the normal infusion of the contrast agent into the body of the subject. But at the same time, it gives you an idea of how good is your system if you were to compare to different systems to detect the sensitivity in that model particularly. And another last example will be to consideration about background and the contrast agent or probe kinetics. So here, this is an experiment that Sean published recently, in which he observed like the Primovis clearance in a healthy animal over 1 hour post-administration. And as you can see, usually the endogenous OATP from the liver and the animal will give you the uptake, signal uptake in the liver, but it clears after 1 hour. So this might give you an idea of like in what time frame you can start imaging your cells of interest that might be in the body of the animal without having too much interference of endogenous signals in the test subject. And the idea here is to keep a contrast enhancement kinetic like always separated in between your test or engineered cells versus non-engineered cells. And this, you can do this by dynamic imaging. And as long as you normalize all the signals to tissue that is basically the background for you, it will be a good assessment. And you just can see actually how early can you start imaging or looking at your images after infusion of the control agent. And this is an example of also from our group that comparing engineered cells versus not engineered cells. If you image at 20 minutes post-contract administration, what you get is basically no difference in between naive cells and engineered cells. But if you wait 5 hours, which we have done prior studies just to determine how long in your system you can be imaging until you see that difference, you will see that the engineered cells are actually distinguishable from the naive or non-engineered cells. So this is something that is important. And as well, last example here is like you have to consider that because these transporters actually are bidirectional, there is an efflux of primovies from the cells. So after 2 hours, 3 hours, the concentration of primovies within the cell and the relaxation rate that you will be detecting will be decreasing over time. So those are things that I consider that might be important at the time when you are actually planning your experiments and the time frames for it. So with that, I just would like to go a quick summary. Basically, we see that it's a transition period for this field in which immune cell tracking using reporter genes is still lacking on the field. We see that this is actually going to be useful for complementation with other studies like PET. But basically, you will need always specialized hardware and personal expertise to do that. And of course, there are always the challenge of sensitivity. That's why most people go more to direct labeling, but sacrificing the long-term tracking. And finally, we think that it's that room of opportunity in which if we're to compare different reporter genes side by side within this cell system, like tracking immune cells, it will be good for the field to have that and beneficial for everybody. With this, I would like to conclude and just say thank you. And if you have any questions, we'll be happy to answer them.
So thank you very much to all three of you. This was absolutely fantastic. And I'm quite impressed how many new things I've learned in the last hour. So thanks again. And we are going to have a bit of a Q&A session now. And I've noted down various questions that came via different channels. And so it is, yeah, so I hope you are okay staying with us a little bit longer. Because we're a bit over time. And to do so, I would like to start with a question that is actually a question that goes to all three of you. And we have heard quite a bit in the last hour about cell detection and cell sensitivity. And one thing that springs to mind in that context is that this depends, of course, also of the reporter expression level in each of those cells. And I was wondering if any of you had data or an idea, maybe some unpublished observations about how much these reporters that you are using are actually changing over time in the cells when you do an in vivo experiment. So maybe I can guide this as to start the answers to Georgia. To begin with?
Okay, yeah, that's a very good question. So what for my experience, for example, what I can say that yeah, when you use a lentiviral report vector, it's quite how to say safe. It doesn't affect how to say the immune tumor profile, at least for LUC2 and the click beetle as well. And the nanolook, it's more the substrate what can influence the performance. For the behavior, so the question is more related if the reporter gene can somehow change the behavior of the cells in vivo. Right? Is that if I?
Well, I meant more about, for example, you would put in a reporter gene, you sort your cells, you have it at a certain level, but if it would change over time in expression while it is sitting in vivo, then the consequence might be that you get a high signal from a region where you have highly expressing reporter cells and a lower signal from a region where you have lower expressing reporter cells. But in a sense, it would be the same number of cells. So the larger that variation gets, the less uncertainty you get on the enumeration of your cells. And I was wondering how much the cells are reacting in vivo to this.
Okay, I think it's something to take into account, especially when you use when you select the promoter that you want to use. Of course, if you use a strong promoter, would be most reasonable. How to say? I mean, would be like mainly expressed by the cells, so can be quite reliable. But if you use, for example, inducible promoter, for example, we have a transgenic mouse in at the Erasmus Medical Center for T-cell monitoring, and this mouse has engineered T-cell when they get activated by the NFAT promoter, and in that case, it's quite hard to in some case of inflammation or tumor modeling can be tough sometimes to distinguish activated cells from just normal cells, so just proliferating, let's say, native cells. So in my case, in the case of bioluminescence, what I'm thinking now, it's maybe related to the promoter that can affect the imaging.
Okay, so Rafa, what is your opinion? Do you see cells in your case basically getting rid of the reporter or shutting it down over time, or?
Yeah, like certainly there is the case of promoter silencing that usually happens over time. That's why one of the reasons why you work with the cell lines, you create a cell line, you should avoid several passages of them, like beyond 20 passages usually. You can get a genetic derived from them. And what we see, I think I have seen data, not publishable, but data of some colleagues that sometimes when they have to sort the cells, they get really different population of expressing cells. And when you decide to do these experiments, you either go by the high expressing one, the medium one, or the lowest one, depending on which one. And the way we do this experiment is we kind of try to test the three of them, the three groups, because you actually either do a clonal selection, will be very specific, or you just grab a group of cells that are around the same level of expression and work with them. Mm-hmm. But certainly, I think I've seen data, especially not with MRI, but fluorescent data, like fluorescent reporters, that over time, like once you freeze the cells and thaw them, over time you see kind of a decrease either in signal or, and what I believe is just some, depending on the method of engineering. For example, the antivirus will deliver several copies. So you kind of get away with it for a longer time. But if you only need do site-specific engineering, just put in a single spot in the genome, the epigenetics might influence. I think if you're gonna do long-term tracking, and that's what I'm thinking of, you have to consider that where within the time span of your experiment, you have to have demonstrated that your cells have the same level of expression somehow. So I guess you can do mid-time experiments, tests using getting the cells and do, for example, flow cytometry and detecting how much level of expression at that time point you still have. Because the decreasing signal, of course, will be not related to the technique of imaging itself, to the substrate you're using for imaging, but your cells are actually giving you a difference, and that might be actually influenced by the response of the cell. I mean, I'm thinking of the immune cells, right? They will change their phenotype over time, but that's kind of my idea. But I haven't seen concrete data or publication actually stating explicitly this is the problem we're having, and I assuming that it might occur.
Yeah.
So maybe this is an area where we in the field need to put a little bit of attention to see how big this possibly uncharted or largely uncharted arena actually is influencing the results, which is of course of only a big impact if it comes to immune cell tracking, as when we think about therapeutics that we want to track in animal models also.
That's right.
And
yeah.
I mean, in that context, of course, basically the essence of what you both say is, well, everyone needs to test and needs to check this. So I can see how that relatively in a straightforward way would work with bioluminescence reporters. But I would think it is rather complicated actually with the MRI reporters, isn't it? Because the function of the reporter, you always have to go to the MRI. Or maybe could you comment on that, Rafa, in the sense of whether for some of the reporters that you are using, you have alternative substrates that are sitting in other modalities, like, for example, a fluorescent substrate for OATP that would make life a little bit easier for doing these checks in vitro?
Yeah, like from what I haven't seen, there are what is it? There is a cyanine fluorophore that indocyanine that also goes through ATP. That has been published already. I think in our hands we don't use that very often. But the thing is, like, it depends on the reporter. In this case for ATP, if you have the chance to use another substrate that can go through it, that isn't seen in another modality, I think that will make cheaper those kind of tests and quick assessments, rather than just going to the scanner. As we talk about, the scanner is another level of complexity and expenses. But for sure there are like that one and the cyanine green, and there are probably a couple more that I'm not exactly remember the names, that because is against the nature of the transporter is not as specific as you might think. So if you have that, it helps in the sense of, okay, I can use another substrate in another modality, or maybe engineering some of the substrate that you already not having one modality with fluorophores, for example. Maybe you can create a system, quality control system for that. And it's actually one of the ideas in our group always is like, before going to the scanner, we have to make sure our cells are properly engineered, and we need to make sure that it's functioning properly. So that's I think it's important. And we do it with other modalities as well.
Yeah, I think that's a very important thing you just mentioned, Rafa, because this is really where obviously the advice from imaging people can actually help others to design the experiments. Because if you use your OATP and would combine it with a fluorophore, then obviously your life gets easier when it's going about this engineering of the cells.
Yeah.
So and so there are two recurrent more questions that are coming from the audience. So one relates to deep tissue imaging with luciferases, and it relates to, in a sense, these questions relate to the body locations. So how big of an issue is it, Georgia, that you have attenuation of the signals, for example, by the rib cage when you look for lung tumors or lung metastases, and what are you doing against misinterpreting the results?
Yeah, that's also a very good question. To my experience, I can say, for example, for imaging the lungs, I did it in the past, and it was quite, let's say, straightforward, because when you inject the substrate IV, the substrate is mainly together with the cells, they are mainly located in the lungs. So this can be very useful to detect also small metastases in the lungs. But of course, from there, the substrate can biodistribute in in the body, and it has to reach the luciferase or the tumors, and that also depends on the substrate itself, because some substrates, they don't have how to say, good solubility. So also take into account where is your metastasis, for example, or where is your tumor of interest. But yeah, so what I can, for example, when I in the past applied the spectral mixing, I was able to distinguish macrophages and metastases. Of course, you can rely on the spectral mixing, but you need to have a certain amount of photons emitted by your system to how to say, to educate your algorithm to distinguish that system. I don't know if I'm clear with that. So of course, if you have, if you already see in a subcutaneous model that the algorithm cannot distinguish your system because already the photon flux is quite low, I won't go for a deep tissue imaging, because yeah, this can, in this case, maybe especially if the two populations are co-localized, one spectrum can overlap the other spectrum that is quite lower. So also the expression of the two luciferases has to be at the same, more or less at the same level. Otherwise, also in that case, can be can be quite tricky to detect and distinguish the cells in the body. Yeah.
Okay,
so,
I see. And sorry, Georgia, and then the last question that we would like to cover here, before we switch to the social launch, which was on the slide from Rafa, is a question, a more general question that relates to the various substrates that all of you guys are actually using for picking up your reporter expressing cells. So are there differences in diffusion, or how long does it take, and how dependent are these substrates on the different areas? And people also would like to know if you have tried imaging in the brain, for example, and whether your reporter genes will actually allow that. But maybe we could start with Rafa and Sean to comment on this. And Sean, maybe you could also say something in relation on whether the T1, T2 signals are actually different in the different body locations for the substrates that you're using. That would be really interesting.
Sure. Yeah. So I think there were a couple of different parts to that question. But starting with the first part, looking at different tracers. So essentially with OATP as an example, if you want to get more agent into the cell, really what you're trying to do is vary that amphiphilic character that I was mentioning, because that's really how OATP is acting. So the first chapter of my thesis is about testing 5 different manganese-based contrast agents. And essentially what we did is we varied the lipophilic side chain on each of those molecules just to try to see whether or not we could promote higher uptake through OATP. So that's definitely one factor. Like, you can vary your chemistry to just physically get more contrast agent into the cell. Those agents will also have different relaxivity, as I mentioned, which is a function of concentration, of course. So really what you want to be doing is getting kind of the most amount of probe into the OATP engineered cells as possible. So that could definitely vary with location. For example, if you did have engineered cells in the brain and your probe was not able to cross the blood-brain barrier, or not able to cross very efficiently, you might not be able to get your probe in high enough concentrations within your cells to get a big T1 shortening effect. So it definitely will play a role into kind of where the cells are and how easily your probe can get there, for sure.
So Rafa, what are you saying in relation to getting them into the brain? Is that a possibility? Does it work?
Yeah, well, I don't have experience working in brain imaging, but I do know that, for example, if you're imaging tumors in the brain, because of the very nature of the leaky characteristic of the blood, the veins and the arteries that those tumors will have, usually this is actually what sets apart the tumor from the normal tissue, right? So I'm not really familiar with brain imaging and using, for example, Primovist, but I think that I don't know, maybe if it's able to cross the blood-brain barrier. I know the gadolinium itself, it will, but I'm not sure if the Primovis. I don't know if Sean have any comment on that, what he's in the literature with that.
So Primovis, like clinically, Primovis is used as a liver targeting agent, so it's not generally seen as kind of something that's making it to the brain. But other non-specific contrast agents like GAD-DTPA, so the same Primovis molecule without the ethoxybenzyl side chain, like that will make it into the brain. So a lot of contrast enhanced MRI done in the clinic is done with that agent. But I don't know if we can touch on kind of in our hands whether or not we've seen that, because I don't believe we've tried brain imaging yet. That is something that we're interested in doing next. I think one of my future projects is going to be looking at that, trying to use OATP as something that allows us to monitor treatment differences in mice that are receiving immune cells. So that'll be something that we look at in the future for sure.
Okay, Georgia, did you try in Rotterdam getting things into the brain, and what are the different properties of the luciferase substrate? So does one have to be really careful because they have totally different tissue penetration?
Yes. It's a good point. So I never worked with brain imaging myself, but Laura has done my supervisor, Laura Mezzanotte, has done it in the past. So far, what I remember is the seq look, that is the one substrate that has been optimized for imaging the brain, but it resulted with a pure solubility. So this can affect, of course, the imaging. I don't know for how long you can image the brain with the seq look. I never tried it, but the poor solubility makes me thinking that maybe the biodistribution won't be that great in all parts of the brain, for example. But I have also made an example on nanolook with cephalofurimazine that has been released like 2 or 3 months ago. They made this cephalofurimazine that is an analog, and it's quite looks like quite promising for improved brain imaging with a very bright emission of bioluminescence. But I must say, in general, when I work with the nanolook system, the stability cannot go more than, the stability, it's a bit variable, so it depends on the imaging that you are doing, and usually it's a kind of a glow type luminescence, so maybe it's not stable and steady over time, but can glow for 30 minutes, 40 minutes, and then it goes down like by half. That's what I noticed, not in brain imaging, but that's what I noticed with the nanolook systems. So something also to take into consideration.
Okay, so thank you all very much for your deep insights into these things. It seems, at least to me, as if there are quite a few things still to be researched. I think we all have learned a lot, also from the discussion, keeping in mind all of those things in relation to the reporters and how long they're there, the efflux and so forth. So I hope everyone who has been listening has got a good insight. Certainly we did here as the chairs. And thank you very much for volunteering for this again, and to share all of your knowledge. And I would like to encourage everyone now to switch to the social lounge. And there we can have more conversations and discussions about the various aspects. But with that, we will close this ICIT organized webinar in the name of the World Molecular Imaging Society. And I would like to again thank not only the speakers, but also the audience for attending. Goodbye.
Thank you. Bye.